Hi everyone, this is the second part of Chapter 3. This is going to primarily cover the process of prokaryotic reproduction and a few more items that we're going to find in prokaryotic cells. So tuck on in, we are going to get started. Now, if you remember anything from previous biology or biology-oriented classes, you'll remember potentially mitosis. And those are the phases of cell division in eukaryotic cells. Binary fission is going to be what we call cell division in prokaryotic cells. This is going to be a form of asexual reproduction because those prokaryotic cells are going to be individual organisms, right? So this is the actual reproduction. It's not just a new cell being created, it's a new organism being created, okay? So this is the most common form of reproduction in prokaryotes. It's also going to occur in single-celled eukaryotes as well as some multicellular eukaryotes, but that's not what we talk about in this class. So how this works is a single parent is going to make a copy of itself into its offspring. So the parent cell is essentially going to split into two halves to make a new cell. There are three main steps that I want you to know in binary fission. This is the first one, elongation and chromosomal replication. I have it labeled out here in that green box to the right. This is going to happen in the parent cell, where the parent cell is going to replicate its DNA. And in this process, the cell itself is going to also double in size in order to hold that extra DNA as well as prepare to split. So this is step number 1 of 3. Step number 2 of binary fission is going to be septation. This is going to be the formation of a partition between what will become two new cells. So this is going to be called the septum. So that septum is that partition between what will become the two new cells, but they haven't formed just yet. So the makeup of that septum is going to be a series of proteins and peptidoglycans. You're going to notice that that DNA is also going to make its way one into one side of the septum and one copy onto the other side of the septum as well, in preparation to be dispersed into the two cells. Naturally, the third and final step of binary fission is going to be the cell separation, where that septum is going to officially split due to those proteins constricting, kind of like a bunch of rubber bands around a watermelon, if you've ever seen some of those videos. It's just going to cut that elongated cell in half and create those two cells. Now, an important thing to note is those two cells are now genetically identical to each other. They're also genetically identical to the parent cell. For the most part, prokaryotic cells are going to use this process of binary fission to create new cells exclusively. But there are ways for prokaryotic cells to be able to share genetic information with each other, so from cell to cell, and how they do that is using these structures called plasmids. Plasmids are going to be self-replicating extrachromosomal DNA. They're a little bit smaller than the chromosomes found in a prokaryotic cell. They contain 5 to 100 additional genes. They are still circular like the chromosome found in prokaryotes. and they are double-stranded. They are going to be used in the process of horizontal gene transfer known as conjugation, which we will talk about next. There are 3 types of horizontal gene transfer found in prokaryotic cells. Those 3 types are conjugation, transduction, and transformation. The goal of each one of these is for DNA to move from one cell to another. It's just the how that changes. So let's jump on in. Conjugation is going to be that first type of horizontal gene transfer. You can think of this one as being a more purposeful gene transfer, where a bacterial cell is going to have one of those sex pillars project out from one cell to connect to another cell. Basically, it makes a bridge so that plasmids can move from the cell with the sex pilus to the recipient cell. In that connection and that transfer, the donor cell is transferring that information, that genetic information, as well as the ability to have a sex pilus to that new cell, that donor cell. So usually this genetic information is going to be decently beneficial to the recipient. Sometimes it can be kind of useless, at least to us, upon observations, usually useless information, but it could be something like being resistant to antibiotics. It just depends. Transduction is going to be the second type of horizontal gene transfer. You can think of this as accidental gene transfer via a virus. Specifically, it's going to be a bacteriophage, which is going to be a virus that infects prokaryotic cells. So they're viruses that infect bacteria. So what happens is when one bacteriophage jumps from one host cell to another, it often steals some of the first host genome in order to make copies of the virus itself, which are called phages. So when these new phages go to another bacterial host cell and infect it, they are taking by accident the genome of that original host bacterial cell to the next host cell, and then inserting that genome into the next host. So again, this is kind of an accident of gene transfer, and it's going to happen due to a virus known as a bacteriophage. So transduction. Last but not least is transformation. This is going to be a very odd one to think about, but it's more of a recycling of genetic material where a cell, a recipient cell, is going to pick up DNA fragments from a dead donor cell. So a dead cell is going to break down, which is called lysing. And then the live cell is going to come along and kind of hoover up some DNA fragments and maybe find something useful, maybe. You can kind of think of these as being a little bit like Frankencells, because it's picking up essentially dead cell DNA fragments and then incorporating them into the live cell, which is kind of interesting. So this is transformation, and that is the last type of horizontal gene transfer. Other things that we can find in prokaryotic cells include ribosomes. The term ribosome should sound familiar if you've talked about eukaryotic cells. You see ribosomes pretty frequently in talking about eukaryotes, but we also find them in prokaryotes as well. It's something that they both share. Remember that ribosomes are going to have everything to do with protein synthesis. So there are two parts to ribosomes. There's a large subunit and a small subunit, and I do want you to know the size of these subunits. The unit that you can use for these guys is S. It's just a form of density. So the larger subunit is 50S, while the smaller one is 30S, giving these ribosomes found in prokaryotes the density of 70S. So what's kind of interesting is that in eukaryotes, ribosomes have a density of 80S, except for in mitochondria and chloroplast, which is more closely related to the ribosomes found in prokaryotes, which is 70S. Because remember, mitochondria and chloroplast have their own ribosomes, their own DNA, which is circular, and this also helps in supporting the theory of endosymbiosis, which we talked about in chapter 1. Another structure we can find in prokaryotes are called inclusion bodies. These are going to also be known as granules. They are found in the cytoplasm of the cell. They're never enclosed in a membrane, and they can serve as a storage vesicle that respond to the environment, such as being able to store food, gas, iron, or other items. They can be a variety of shapes and sizes, and that's a really important feature of inclusion bodies because the next structure we're going to talk about is very similar but different. Bacterial microcompartments are going to be very similar to inclusion bodies since they are a storage unit, but they are a specific shape. So they have to have a specific item that they store. They specifically store enzymes, so proteins that catalyze reactions and work for a variety of biochemical pathways, if you remember what an enzyme is. Remember that enzymes are going to speed up reactions, so these are just going to be compartments that hold those enzymes to help with those processes later. The shells are going to be made of proteins that are arranged geometrically to specifically hold that specific shape of enzyme. This moves us into prokaryotic sporulation. So this is going to be the production of spores and endospores. We talked about this a little bit in chapter 1, part 2. So let's go into what an endospore is, what it does. Remember that some bacteria can produce an endospore. When we were talking about it in chapter 1 in relation to Ferdinand Cohn discovering them in the 1870s, these can be produced from bacteria in tough environments, or if the environment changes to where the bacteria is either going to potentially die, it may produce a spore if it's able to. And that spores are kind of like an egg, but not really an egg. We'll talk about it. So in relation to endospores, the production of a endospore is called sporulation. And again, this is going to happen when the environment becomes harsh, or maybe the bacteria is lacking in nutrients. It just depends. When the environment returns to favorable conditions, then the bacteria is able to return to its normal reproductive state, and what we call this is germination. So, a couple examples of bacteria that are able to create endospores are certain gram-positive bacteria that maybe you've heard of, including bacteria in the genus Bacillus and Clostridium. An example that maybe you've heard of in the medical field is C. diff. We'll talk about that one quite a bit actually in this class. A couple examples of endospores in recent advancements or recent studies. 5 years ago, we recovered some bacteria from an ice core in Antarctica that had bacteria that was 300 million years old. And the reason that that bacteria was able to survive that long in the ice is it had produced endospores. And once that bacteria was released from the ice, those endospores started to germinate. And yeah, so super scary. We immediately incinerated those little bad boys. And it does help that they were preserved in ice, but the endospores definitely allowed for that bacteria to survive for that long in that condition. So this slide shows a nice overview of binary fission and sporulation. It breaks binary fission down into a very simplistic process. I do want you to know those 3 steps we talked about. I do not need you to know the steps of sporulation. More than anything, this is just kind of an interesting process. Again, I told you spores were kind of a little bit like an egg, which it is. So we can see there at the bottom that that spore is being formed, and it's kind of preserving, in a way, the bacterial cell or the cell in general, and then germinating to create that cell so it can reproduce normally. But if you want to think of it this way, those spores are going to cause the cell to become dormant for some period of time until the environment is favorable again. But again, you do not need to know the process of sporulation, just what it is, and then those terms, sporulation and germination, what we were talking about on that last slide. But you do need to know the process of binary fission. Now remember that bacteria is not the only type of prokaryote. So we are moving away from bacteria, but we are still talking about prokaryotic cells, which is the domain Archaea. These guys are going to share a lot of characteristics with bacteria, but they are decently similar to eukaryotes, a little bit more than they are to bacteria. But they are still prokaryotic in their cell structure. They are still a very primitive form of life. They are unicellular, so they have they're single-celled, and they're most closely related to the first cells that originated on Earth roughly 4 billion years ago. We know this because modern archaea are going to be something called extremophiles. Extremophiles are going to be those types of organisms that are able to live and thrive in really extreme environments. And I'm going to have a list of the different types of extremophiles that I want you to know on the next slide. And the reason why we're talking about extremophiles and how archaea are really closely related to those primitive life forms, is because that early Earth, remember, was essentially a hellscape. So the fact that these guys thrive in extreme environments, extreme heat, extreme acid, no oxygen, etc., or really salty environments, it really makes sense that archaea are really primitive life forms because they came from organisms that were some of the original organisms of life on Earth, because that's when Earth was really extreme in its environments. So let's talk about it. When it comes to our types of extremophiles, you have temperature extremophiles. There are three, I want you to know. There are thermophiles. These guys are going to live at temperatures that are a little bit on the warmer side. So they are known as heat lovers. So thermo being heat and phile being lover. Hyperthermophiles are going to be extreme heat lovers. You don't need to know the temperature ranges, just know the term and maybe in general what it means. And then we have psychrophiles. These guys are going to be our cold lovers. So those are our three temperature extremophiles. We have an acidophile and an alkali phile. These guys are going to, as per their name, either prefer more acidic environments or more basic environments. So we have our acid lover and our alkaline lover. An example of an acidophile is going to be an organism that we talked about before, Helicobacter pylori, which is a bacteria that lives in stomach ulcers, which, you know, if it's in the stomach, that has a pH of 2. So it makes sense that those guys are acidophiles. You can have anaerobes. These guys do not require oxygen to survive. There's actually quite a few microorganisms that are anaerobic, so they don't need oxygen in their environment. You have halophiles. These guys are salt lovers. You can think of microorganisms that will survive in the Dead Sea. If you don't know what the Dead Sea is, I wish I had a picture up on this screen. That'd be kind of cool. But it's essentially a very salty sea or salty body of water. And it's kind of shocking that these microorganisms are able to survive at those salt levels, but they're extremophiles, so that makes sense. You have osmophiles. These guys are, instead of being salty, they're sweet. They are sugar lovers. And then xerophiles. These guys are going to prefer really dry, desiccated conditions, so no moisture. As of right now, Archaea have not been linked to causing human diseases, although more recently we have found some Archaea microorganisms found in gingiva of people with various levels of periodontal disease. So at some point, we could start finding links to these types of microorganisms causing or being linked to human disease, but as of the moment, we're not really seeing that. It could be more like they just kind of were there as far as the gingivitis, but who knows? Who knows? New information is constantly being pumped out. We're constantly finding new links, new information. That microbiome, human microbiome project is really important. So who knows? We might be finding new information within the next couple of years linking Archaea to various diseases. So the way we classify prokaryotes is going to be very similar to how we classify eukaryotes. We talked about that at the end of chapter 1. There are 2 databases for classifying prokaryotes, and they're based on 2 different features that we can compare prokaryotic cells against other prokaryotic cells. The first is Berge's Manual of Systemic Bacteriology. This is going to use RNA sequences. It's going to be incredibly accurate to compare species of prokaryotes to other species of prokaryotes, but it's substantially harder to do. The second is Berg's Manual of Determinative Bacteriology. This is going to be based on morphology and phenotypic or observable characteristics, as well as metabolic capabilities. So this is pretty useful for students of medical microbiology, but it's not always as accurate as comparing the RNA sequences. Now, that systematic bacteriology is going to organize prokaryotes into 4 major categories. And when we put kind of an image to these terms, these categories as well, then you're going to be like, oh, cool, that makes sense. We saw that before in Chapter 3, Part 1. So, the first one is gracilicutes. These are going to be bacteria with gram-negative cell walls. They're going to be considered thin-skinned. Next is firmicutes. These are bacteria with gram-positive cell walls. These are going to be thick and strong-skinned. Tenericutes. These are going to be bacteria that lack a cell wall, soft-skinned. And mendocicutes. These are archaea with primitive cell walls, no peptidoglycan, and nutritional habits. So, when it comes to this last word or last part of the words that I just gave you, cute is meaning skin. You can think like back to your A&P days, like subcutaneous, right? So it means skin. So let's get some images up on the screen to help pull this together from before. So these images should look very similar to the images that I showed you when we talked about gram-positive and gram-negative, as well as looking at the layers of the cell wall of prokaryotes. So this is in relation to that Berg's Manual of Systematic Bacteriology and how it relates to those terms that we were just talking about. So the gram-negative bacteria is going to be our gracilicutes. Remember, that's going to be the thin-skinned, which makes sense because it does not have that thick layer of peptidoglycan. The firmicutes, which are going to be our thick and strong gram-positive bacteria, they have that thick layer of peptidoglycan. Then our tenericutes, which are going to be bacteria that lack a cell wall. They're considered soft-skinned. They don't have peptidoglycan. The proteins within the membrane prevent lysing, but they're very fragile. So these are our mycoplasmas. Again, this is still bacteria, but they lack a lot of the structures we were just talking about with gram-positive and gram-negative. They are neither of those. And then we have mendocicutes. So these are going to be our archaea. So these are those primitive cell walls, no peptidoglycan, and no nutritional habits. So normally the species level is going to be the smallest, most distinct level of classification that will label a species as a particular organism that can produce viable offspring when it mates with its own kind. But this isn't true for prokaryotes because they use the process of asexual reproduction or binary fission as its form of reproduction. They do not mate with each other like a cell does not mate with another cell. They reproduce asexually. But they also have horizontal gene transfer. In some cases, some bacteria can have subspecies. This is bacteria of the same species that have differing characteristics, and also sometimes called types or strains, which we will talk about different types and strains of bacteria. We've already talked about this a little bit with E. coli, that we have a particular type of E. coli in our guts, but there is one known strain that can cause massive food poisoning if your food is contaminated. So that is a strain of E. coli, just as an example. And with that, guys, that is the end of chapter 3. I hope you enjoyed all of this fantastic information about prokaryotic cells. Chapter 4 will be all about eukaryotes, and it's a decently short chapter, but I hope you stay tuned and enjoy it. And remember, make good choices, guys, and make sure to study. And if you have any questions, you can always email me.